Hello, everyone. Welcome to Global Health Watch. This is Dr. Sean Ling. For the last few days, many people's attentions are on California in terms of COVID-19's outbreak, because in California, especially in Los Angeles County, Santa Clara counties, 1/3 of the population was actually testing to be positive. So the cases of positive COVID infection has increased tremendously in California. And of course, many people also saw the news today that even the Californians has sent out an alert for people who prepare to be vaccinated by Moderna's vaccine. Right? Moderna has this mRNA vaccine supposed to deliver hundreds of thousands of doses to California. But in the last couple of weeks, because unusual high numbers of allergic reactions for the people who vaccinated for this Moderna vaccine, so California's Department of Public Health decided to temporarily suspend the distribution of this vaccine. So many people worry about the situation, right? On one hand, you have a significant increase of positive case. Seems like the virus being spreading out fast in the community. And then at the same time, government actually have to temporarily suspend the Moderna vaccine. But of course, you can try Pfizer's vaccine, but people worry about, will I also get side effects from Pfizer's vaccine as well? And then the bigger question is, how about the virus mutants that emerged in United Kingdom, emerged in South Africa? Did it pop up in California? And last week, we also heard about the mutation has been identified in Ohio. 2 different new mutant strains emerged in California, in Ohio. So what about California? So in today's program, I want to talk about the mutants that have been identified in California. Right? So let me show you my screen here. Right. So in California, many people have been lined up to do different tests in California. And also California definitely have strong virus genome sequencing capability, especially in UCLA's, UCSD's. And so in general, the sequencing capability in California was much higher in many of the states in the United States. So they have been able to identify new mutant emerged. So in here, I'm presenting to you a tweet from Dr. Charles Chu. He's from UCLA and UC San Francisco. Yeah. So his lab had been identified a mutant called L452R mutants. Let me highlight this one for you. So this is the L45R mutant, and these mutants also happen in the spike proteins for the SARS-CoV-2. And you can see from the diagram below that even in December 13, at the time, in California, you only see 7 cases of L45R, right? Only 7 cases. But by the beginning of January 3rd, they have identified 37 cases. So this is a jump from 3.8% to 25.2%. Just less than 2 months, it has significantly increased of these new variants presence in California. Right. Let's look at the next slide. And I was searching around trying to find out the detail of the mutants, but this is so far what I can get because from California's Department of Public Health public announcements, as well as Santa Clara County's announcement, we didn't see the details of the mutations. And Dr. Charles Chu has now published his epidemiology study yet. So I only find out this tweet from Dr. Emma Hotcroft. And in her tweet, she pointed out that the mutations, 3 mutations that Dr. Chu talked about is actually 3 in spike. And so definitely the L452R, that's lysine to this mutation L452R mutation, and then you also have serine 113 to isoleucine and tryptophan to 152 to cyanine mutations, and this has been making headlines today. So these all in spike protein, and S13I is at the N-terminal domain, and W152 is also at the end of the N-terminal domain. So these are important regions that can cause virus change its conformation on the spike proteins. So let's look at the next slide. So people, of course, will ask the question, does this L452R mutants actually related to the B117 mutant identified in the United Kingdom? But actually, they are different, right? And we will show you more details about the different mutations in the B117, so you can see the clear difference. And in here, we want to highlight from one tweet from Alexander Bose, and he mentioned that actually 4 mutants of B117 has been identified in the United States, and 3 from California and 1 in Florida. So in California, it definitely has these mutants from the United Kingdom, B117. And now on top of that, you have the new L452R mutants. Right. So next we will see so this growth curve about confirmed case of COVID-19 in California and Florida show you some interesting different patterns, right? You see a significant jump of the positive case in California compared to Florida. So of course, both states have significant increase of these COVID positive cases. But why California has a significant drive, right? And from previous slide, we see Florida also have 1 case of the B117, and then California have 4. But so the significant increase in California was due to the B117 being spread out faster in California. That may be the reason, but I think one of the reasons that people suspected right now could be due to the L452R mutant, because this L452R mutant is immune escape mutant. It may get more people sick, and then when more people getting sick, then they have higher chance to spread the virus to other people, right? Because if people are getting sick, they will have more virus in their respiratory tract, so it can be easily to transmit to the other people. So in here, I want to introduce to you about 2 studies and regarding this L452R mutant. So we need to keep in mind, so this L452R mutant is not first identified in California. Actually, last year it was identified in other countries as well, in other states in the United States as well as in California. So just the L452R mutant is not totally new mutations. But however, when this mutant combined with other mutations, it may present different threat. But in here, I also want to talk about these 2 studies, because these 2 studies emphasizing that L452R mutant is actually an immune escape mutant. So the first study I want to show you, this study is from scientists from Seattle. So it's in the Fred Hutchinson Cancer Research Center. So from Dr. Bloom's, Jesse Bloom's group. So their studies using the yeast expression system to screen mutants, and at the same time they also identify the naturally occurring virus mutants, and they try to sequence these mutants, identify the mutation, and they can pin down from many of the sequences that they've been identified, and then see how many of them actually escape the polyclonal serums. So in this study, they actually identify 4 most frequent mutations, right? So these mutants, some of you probably already familiar with the N5011 mutant that's been identified in both the mutants in South Africa as well as in United Kingdoms. And we talk about the E484K mutant in my previous program. And in here, we want to highlight the L452R mutants. Even though the mutation at these key sites has less frequency compared to E484K, but clearly it's still a very important mutant that keep popping up, right? So let's clear this and move it to next slide. So in this slide, we can see the L452 mutant is on the surface of this spike protein and is critical for the conformations for spike protein to interact with the receptor ACE2. So the receptor here is presented as a ribbon, and while the spike protein is presented as this global molecular shape. And in here you can see the L452 was depicted as a pink color. For dark pink colors, so at least 50 variants count in GSET, and it will showing have a strong effect on the binding. So GSET is the sequence bank that many people who conduct this virus genome sequencing will deposit their virus genomes data into this data bank. So you can see more than 50 variants have been identified to have this L452R mutation. Right? So that's suggesting this mutant occurs frequently, and now it's spreading out more in California. So more of these mutants have been identified in California. And the next study is actually an important Cell paper. And Cell is one of the most esteemed virology research paper journal as well. So let me. So this is study was done by a group of scientists from Beijing. And so let's show you. So basically in this study, they not only identify the variants in the mutations in variants in the circulating situation in different populations, different continents, but they also do pseudo-type viruses. So they put the virus mutation in a spike protein and combine with the pseudo-type virus, and then testing its infectivity with or without monoclonal antibodies, or with convalescent plasma as well. And this study also studied the glycosylation deletion of the pseudotype virus. So this part is not related to today's topic, so I won't talk about it. But just about these naturally occurring variants. So they've been identified several immune escape mutants in their study. And let me highlight this group here. So this is the group for immune escape mutants, and you can see here L452R is one of the key mutation site that can bring immune escape. So this is by incubating the pseudotype virus containing these different mutants with different monoclonal antibodies. Let me show. So on the left axis, on the y-axis, you can see this is different monoclonal antibodies, and for the x-axis, these are all the different mutations that they're screening try to study how it binds into monoclonal antibodies. So in this heat map, for the blue one, it's suggesting it's a reducing of the monoclonal antibody recognition for these spike proteins, and for red ones, it's suggesting these virus actually had enhance the monoclonal antibody with the spike protein. So in here, we want to highlight the L452R mutants. And this L452R mutant is right here. So you can say this one mutation at L45R significantly reduce the monoclonal antibody recognition for the spike protein. So this definitely suggesting it's an important immune escape mutant. Right. And in this study, they also present a table that show you many other mutants that presenting increased sensitivity to the neutralizing antibody or decreased sensitivity to the neutralizing antibody. So here we can show you where the L45R is, right? So definitely L45R belong to those mutations that can reduce the sensitivity to neutralizing antibody. This is suggesting the immune escape, right? So we can also see, very interesting, one of the glycosylation site in the spike protein can also decrease the monoclonal antibodies recognition for the spike protein. So just one additional note here. At the same time, look at this table. When you're using convalescent plasma, because it contains polyclonal antibody against different part of the virus. So in here, we see many mutations can escape, but not the L45R mutations. Right? So this just keep in mind. So L45R alone may not be able to completely render the virus being capable to escape the whole polyclonal antibody or convalescent antibodies in patient's body, but it can definitely interfere with the virus conformational structures by the spike protein, right? So in this way, it can work with other mutations, for example, deletion in the N-terminal or maybe some deletion in the open reading frame A proteins. So it can still make the virus changes conformation, changes function, and make it immune escape mutants. So still a very important mutants, right? So let me look at another slide here, right? So we talk about the L452R mutants, very different from the mutants from United Kingdom, the B117 lineage. So this diagram shows the mutations existing in the United Kingdom mutants. So let me highlight here, right? So you can see the coronavirus spike genes here, and this part is this domain area, this spike proteins gene. And for this B117 lineage, it has several important mutations in this receptor binding domains, right? And they also have an important deletion at N-terminal. And so the new mutants identified in California does not show this deletion yet. But anyway, we still need to wait for California scientists for a research paper to see if they have any other mutations, you know, in, for example, in open reading frame 8 or in open reading frame 1AB, and it may have important amino acid deletion as well. So this part we still need to wait. It's still early to say whether this L452R mutant is definitely more potent in transmissibility or more potent in immune escape. But definitely we just need to provide this alarm to people in terms of these mutations. Right. And compared to the mutations in Brazil, we can take a look at here. This is one of the studies investigated about the mutation happened in Brazil. And you can see these mutations definitely has these cluster of mutations that define this lineage, K417T, E484K, N501Y mutants, and they have this immune escape mutant E484K as well, and also have an important deletion in open reading 1AB, and also have an important mutation in nuclear capsid protein. These will also change the antibodies as well as cytotoxic T cell lymphocyte recognition of these mutant viruses. Right. Okay, so let me see. All right, so this end of my slide here. So basically, I want to point out to everyone that these mutants newly identified to spread out widely in California, the L452R mutant is giving a very important alarm to people in California, because these mutants has increased its presence in many different counties, Los Angeles counties, San Bernardino counties, and Santa Clara counties, and many counties in California has these new mutant emerged. And right now, we, from very limited information, we only know the mutation in the spike protein. But what about other mutations in other component or other part of the genomes? We need to wait for the study. But because it already showing immune escape mutants, the L452R mutation, so it definitely showing these virus has been learning to break through the immune defense system that people have. And if these mutants continue to spread out very fast in California, then it may further adapt additional mutation that maybe bring to even higher pathogenicities. Right now, we know in California, in Los Angeles, in Santa Clara counties, there are more death case people being infected by COVID-19 and then died. And even Los Angeles area, they have to temporarily suspended the restriction for cremation of human body because in California they have these kind of restriction in terms of how many corpse you can cremate, you can burn it, because it will pollute the air. When you burn the human bodies. So the California actually had this regulation restrictions, routinely restrict the number of body that can be burned. But right now, because so many people die from California, they even have to lift this restriction temporarily. So that's suggesting more people dying in California due to the COVID-19 right now, even though there is no comprehensive study to prove the new variants lead to higher pathogenicity, lead to more people deaths. But we need to keep this in mind. Maybe in the near future, we will see this kind of study. So definitely, we need to be really vigilant on the development of COVID-19 situation in California. And especially right now, so many people put the hope on the vaccine, but even Moderna vaccine right now is showing unusual high percentage of the allergic reaction. So, we just need to be fully alert the challenge that we face. And this happened in California, but it may also happen in other states soon. So overall, just really presenting a very concerning situation about this emergence of 452R mutants. So hopefully there will be less cases being imported from other countries, as well less cases coming from California spreading out to different states in the United States. Well, we can only pray right now, because the virus mutates so fast. You can see the vaccines rolling out speed is pretty much crippled at this kind of stage right now. Anyway, that's what we want to present in this program, and I will also talk about the Moderna vaccine, about this mRNA vaccine, why it may have this higher percentage of allergic reactions or adverse reactions based on previous RNA vaccines developments. And so I'll talk about it maybe in next episode. And thank you for watching this episode. And that's all for today. Thank you very much. This is Sean Lin.